טוב, אני רק רוצה לשאול אם יש מישהו שהוא דובר אנגלית, any English speakers in the audience, perhaps we'll switch into English.
הכוונה מי שאינם דוברי עברית?
כן. If not, we will continue in Hebrew.
If you do it in English, that'd be glad.
Okay, so we have a non-Hebrew speaker in the audience. Perfect. Okay, so I think I will start. Let me first introduce myself. My name is Nili Vershov. I'm the head of teaching and students at ELSC, the Edmond and Didi Safra Center for Brain Sciences. Our meeting here today is to introduce the summer internship for this year. And of course, meeting some of our PIs, our investigators. As you can see, Hermona, Soreq, Chaya, Kalcheim, Ben-Yakov, Didi, Omer, Citri, and last but not least, Mickey London. They all will have a short introduction of their lab and of the research. I think I will open with a short clip we have of one of our researchers who is not participating in the open day. Her name is Nomi Habib. So let me just share the screen. Just a second.
In the heart of Jerusalem, researchers are hunting for the answers to aging and brain decay.
Alzheimer's is a neurodegenerative disease, meaning that there are nerve cells, neurons in our brain that are the key functional unit of the brain that are dying. When you look at the individual, you have memory loss and cognitive impairment and mood disorders that are slowly kind of taking away the essence of the person in front of you. And it's really devastating to see.
And an aging population means there are far more cases.
There are estimates saying that 1 out of 3 elderlies in the United States are diagnosed with Alzheimer's at some stage. You look at a textbook on Alzheimer's, it seems like a lot of the mechanism is already figured out. But when you try and translate that to the clinic, it seems like something is missing.
But at the Edmond and Lily Safra Center for Brain Sciences, Professor Naomi Habib is finding out exactly what is missing.
Our goal is to understand mechanisms of aging-related pathologies, and specifically Alzheimer's disease. We're trying to understand how does, on the most basic cellular molecular level, how does the disease progress from a healthy brain to advanced Alzheimer's disease. And our goal is to find these early triggers of the disease so we can find effective therapeutics against them.
We're very interested in looking at cells that are actually aging. We get donations from live brain tissue from surgeries, and we can grow this brain tissue of a human in our incubators as well. So we have the chance not only to look at cells, but to look at actual human cells at different ages from different patients. And then we have the same advantage here, we can manipulate those cells.
And what makes this lab unique is the ability to do those experiments at the smallest scale.
Now we can actually look at individual cells. And then later in the computational analysis, we can figure out who these cells are and build maps of the whole brain. So how is this done? The key point is our ability to actually isolate different cells, capture them, and tag the molecules of each of these cells individually.
The cells that we see in this map, they come from hundreds of different individuals. Using this diversity, we can now try and put all of these donors along some disease timeline, or to try and learn some disease timeline. And we are finding that we have different of these trajectories leading to different disease outcomes.
And that is uncovering the answers to many long-standing questions.
Some people were claiming that Alzheimer's is an accelerated form of aging, meaning that if we all continue to age until the age 150, we will all get Alzheimer's. But in fact, what our maps show is that there are different paths the cells in the brain can take. One of these paths is where all the cells together are changing their functions leads to Alzheimer's. And the other path leads to the slower decline that we associate with more healthy form of aging.
And from the answers in Habib's lab, a path to a cure.
There are people that have pathologies in their brain, but they don't have the clinical symptoms. So these type of resilient individuals, we believe, are really the key to find a cure. So we're measuring the brains of specifically these individuals, comparing them to people who have Alzheimer's, and trying to find what are the natural mechanism of resilience.
And from there, a possible cure for millions.
אוקיי. אז זו הייתה מונה חביב, יש לנו עוד... sorry, we have to switch into English. So this was Mona Habib, of course, and we'll move forward to the other researchers we have here today. Please note that I will answer questions later on. I see that Hermona Soreq is here with us. Hi, Mona. Good morning. Please, if you could unmute yourself.
Good morning.
Perfect. Hi. Good morning.
Hi.
Later on, I will answer questions. Please, let's hear Hermona Soreq.
Okay. Okay. First, I need to tell you that I join Naomi in appreciating the impact of her studies into Alzheimer's disease. And second, in our lab, we do that from a different angle. And what we actually do is to look for small RNAs, not mainly by analysis, but by combining experimental work with analysis. So our concept is that if you think about gene expression as a pyramid, you would see genes at the top, proteins in the bottom. You don't want to hamper with genes because that might entail terrible outcome. The proteins do the job, but to attract a protein means that a lot of other sub-effects might come out too. So we do our work on the middle at the level of RNAs. And over the past 5 years or so, we focus on a small family of RNA fragments that are broken off from transfer RNA, which is known as the mediator that takes amino acids and brings them to build the protein chain. However, what we discovered is that when that transfer RNA gets shredded to pieces, each piece might have a biological activity. If we miss those pieces, we get an outcome of facilitated deterioration of cognitive ability in women with Alzheimer's disease, a specific impact on women, and the mechanism of action is related to the depletion of such fragments of RNA that can have a very important regulatory function. So in our lab right now, we have a student who is focusing her studies on these transfer RNA fragments in the Alzheimer's disease brain. And if you join us, you'll be welcome to ask what are the levels, what is changed, what is modified in that disease. But this is not the only disease. If we look at Parkinson's disease, we find that depletion of particular transfer RNA fragments of mitochondrial origin, again, those coming from our mother, because this is where we inherit mitochondria, is accompanied by an elevation of many fragments with a consensus repeated motif. And Nimrod Madrer in the lab is studying these fragments both for diagnosis and hopefully for future treatment of Parkinson's disease. So these are two diseases. The third is mental disease. And we have a postdoctoral fellow in the lab, Dr. Tamara Zorbas, who looks for the functional impact of these transfer RNA fragments on neurons in culture. And she takes those fragments that accumulate in the brain of schizophrenic patients, which we got samples of from the Netherlands Brain Bank, adds them to cultured nerve cells and sees a change in reaction to calcium ions, so altered neuronal activity. So here are 3 examples: Alzheimer's disease, Parkinson's disease, and schizophrenia. And whoever joins us gets to go through all the students and postdocs in the lab and join the team that best fit their wishes and our wishes. So let me wish everyone a wonderful summer and look forward for you visiting us. Thank you.
Thank you.
Thank you. Thank you so much, Mona. Have a great week.
You too.
So next is Chaya.
Just Neely, if we have questions, so should we ask them at the end, or should we?
If you have a specific, I suggest maybe to email Mona, because
All right.
we want
Perfect.
the
I
rest.
always answer my emails. Yeah.
Yeah, I
All right,
think it's
thank
the
you.
best, because we have more researchers waiting, and I don't want to change the schedule. Okay,
Right.
perfect.
Thank
So if you have questions to any one of the researchers, you can email them, and I will answer questions administration related at the end.
Perfect.
Okay.
So
Hi.
Do you see my screen?
Yes, and we hear you.
Okay, so my name is Chaya, and our laboratory sits in the medical school at Ein Karem, and I am part both of ELSC as well as of the Faculty of Medicine. And in contrast to my 2 friends, Naomi and Mona, our laboratory is interested in early development of the nervous system. So imagine that this is one of the most complicated systems in the body, if not the most complicated one. And we reason that in order to try and understand how function is performed in the nervous system, we would need first to address how the complexity is generated. And this happens during embryonic development. So what I'm going to give you is a very, very general overview with, I hope, nice pictures which come out from our work and the work of other people, trying to summarize what we want to achieve. So basically, the lab works on 2 systems. First is the nervous system. Why can't I move this? Okay, so first it is the nervous system and the accompanying mesoderm and the interactions between them. Do you see my cursor? Do you see it? Okay, so the nervous system develops from a germ layer in the embryo that is called the ectoderm, and pretty soon after specification into becoming a neural progenitor, these cells undergo a kind of invagination, and then the folds approach each other and close themselves to form a tube. So you imagine a baguette in which you have a closed tube running into the plane of the screen, and this tube that you can see here in this scanning electron micrograph is going to be the progenitor structure of the spinal cord, of the brain, of the entire peripheral nervous system. And what we see are very important interactions between these midline structures. This is the neural tube at the level of your spinal cord, and the adjacent mesoderm, these little beautiful cubes that will develop into muscles, vertebra, dermis, and other derivatives. Why is this important? Because, for example, if you imagine motor neurons, they have to leave, their fibers have to leave the spinal cord in order to reach the muscles and create neuromuscular junctions. So a very intimate talk has to take place between mesoderm and the neural progenitors in order to pattern normally the way the nervous system is going to function. And the same occurs towards the dorsal part of the neural tube, which generates the sensory domain of the central nervous system. And this is generated first by these beautiful cells that I hope you can see here, these beautiful little cells that leave the neural tube and will generate the entire peripheral nervous system. So we work precisely on these 3 stages of dorsal nervous system development. The first is called the neural crest stage, in which a very interesting, amazing, I would say, population of cells, which is born in this aspect of the neural tube, will leave the neural tube to create the entire peripheral nervous system. After they stop emigrating, these cells will form what is called the roof plate, or the roof of the central nervous system, which in itself, as you can see by these arrows, promotes via certain morphogen and growth factors the development of additional cell types here called dorsal interneurons, those that are responsible to capture information from the periphery, temperatures, pain, touch, proprioception, namely our sense of where we are in space, and so on and so forth. But this is not the end, because days later, these roof plate cells will elongate and generate a spaghetti type of cell type, which are called radial glia. And these radial glial cells act to behave as a kind of block in the midline for axonal crossing. So cells born in this side will not be able to send axons there, thereby generating the right-left asymmetry and independence of our body. And in addition, these radial glial cells in adulthood behave as stem cells that will generate astrocytes and neurons after injury, and maybe also during homeostasis. And this portion of the tube is very, very important, because if we go to the brain area, namely at these levels of the axis, this will generate, for example, the choroid plexus that synthesizes and generates the CSF, the cerebrospinal fluid, and so on and so forth. So let's see what are crest cells. These cells are marked here by a GFP encoding DNA, something up. I didn't want to do this. I wanted to show you how these cells emigrate from the neural tube and undergo this gorgeous epithelial to mesenchymal transition. We are able to follow these cells in vivo and in vitro, learn their characteristics. And during many years of work, my lab has discovered the molecular mechanism of emigration of neural crest cells. And we know how roof plate cells, those cells that are blue here, reach the dorsal midline after neural crest cells leave this domain and form sympathetic ganglia, Schwann cells, dorsal root ganglia, and an additional amazing derivative, which are melanocytes. All the pigment cells of your body come from this amazing structure called the neural crest, and indeed many neurocrestopathies, many diseases of the nervous system also involve aberrant and abnormal melanogenesis. And of course, I don't have time to enter into this. So basically, in our lab, we are trying to use avian models, quail and chick embryos, which are amazing models for microsurgery, for genetic manipulations, for in ovo manipulations via what we call electroporation. We put the embryos in what I call an electrical chair. Embryos sit within the egg, and we add electrodes, and we can based on these electrodes, misexpress many genes. Of course, I don't have time to show you that, but for example, look at a neural tube that has been electroporated here with a control plasmid, and another one which has been electroporated with a dominant negative plasmid that inhibits the activity of retinoic acid in this case. And what we see is that cells come out very late at the stage in which they should not come out, and these very abnormal cells co-express markers of glia and melanocytes, something which is completely abnormal in the embryo, and we are trying to understand the underpinning of these events. So basically, these are our embryos. They are not more than 2 to 3 millimeters in size. All our work is being performed under microscopes or binoculars. So the people who join our lab should have a very stable hand and a very good communication between their brain and their hands. And during the past few years, a part of our molecular and cellular studies, we are also performing single-cell RNA-seq, which is trying to uncover the transcripts or the RNA species that are expressed in every single cell in this dorsal area. And we are making a lot of progress in trying to understand how neural crest cells end their emigration process. How do they transit into roof plate cells? How do they transit then into radial glia and ependyma? Moreover, we study a little bit of the ventral nervous system, the relationship between motor neurons and muscle cells, and how do we get motor neurons from progenitor cells. So basically the lab works on neural development at the normal level, bearing in mind that this is a prerequisite for understanding further patterning, function, and diseases of the nervous system. So whoever joins us may join us either in the skeletal part of our investigation, together with 3 students working on that, or on the neural part with 4 additional students working on different aspects of crest, roof plate, dorsal interneuron, and motor neuron development. So you're welcome to visit the lab in any event, and I wish you the best of luck in whatever you choose to do.
Thank you so much, Chaya.
Thank you.
So after we know what baguette and spaghetti means at Kalcheim's lab, we'll switch to Ben-Yakov.
The baguette is because my postdoc was in France. My husband is French. We like baguettes, and the baguette is a fantastic example of how a tube looks like.
It's amazing. Thank you so much.
Thank you, Nili.
Toda, Shavua Tov. Bye.
Bye, Shavua Tov.
Aya, hi. Good morning.
Hello. Good morning, everyone. Should I just start or?
Yes.
So we're going to have quite a switch to a very different topic. I'll tell you a bit about the work in our lab. In general, it's a lab looking at human memory. And our goal is to search for the basic unit of episodic memory. What I mean by that will become a bit clearer as we go along. So first, it's important to say what I mean when I say episodic memory. And for this, I'll enlist inside out. So this is an example of what I'm talking about when I say episodic memory. Now, this is maybe because I have young kids at home, but in general, it captures a lot of our experiences in real life. But in lab settings, typically the way people studied episodic memory was looking at very brief, discrete elements. So an episodes like I showed you would maybe be shown as a sequence of pictures. And in this case, it's meaningless to ask what the basic unit of memory is. We save the chair, then we save the broccoli. But when we have continuous experience, this question becomes much more relevant. And the hypothesis we have is also very nicely demonstrated actually by Inside Out, what we think is going on when we encode naturalistic experience. So in the movie, we have these episodes, and we see that at the end of every episode, it's encoded in kind of an orb or a capsule. And this is what we think may be going on in our real life experience. And what led us to ask this was an fMRI study run quite a long time ago, where we presented participants with brief scenes from films in the scanner. And we later tested their memory for this. And we then divided the clips into those that were remembered versus those that were forgotten. But instead of doing what is typically done, which is to look at the brain activity during clips that are remembered compared to clips that are forgotten to find regions in the brain that are involved in encoding to memory, we looked at what happens at the end of the clip. And we found that the hippocampus, which is the memory region, actually waits until the clip is over. We see here the average response in the hippocampus. And if I cover the duration of the clip, this is the response at the end of the clip. Now we see a higher response for remembered versus forgotten clips. And this is very similar to what we see when we measure BOLD, which is what is measured in fMRI. We see a stronger response. I don't know if you can see my cursor. We see a stronger response, and you can see that if we compare it to the typical response, it looks like the response in the hippocampus is occurring at the end of the clip. And to further test this, we had clips of different lengths, and we find that indeed the response in the hippocampus is later for longer clips. That is, our main region of encoding to memory waits until the event is over and only then responds in a way that's sensitive to memory. So this could be encoding of the entire event as a whole. And this led us to this general question of asking how we encode dynamic events that unfold over time to memory. It has three parts. How do we segment these events? How do we encode them to memory? And how are these events then represented in the brain, including this unfolding over time? So the segmentation, just to give you a taste of what we're interested in segmentation, we ask, when does one event end and a new one begin in real life memory, which is continuous? Specifically, we look at things like prediction error, which is failure of our predictions of what's going to happen, and sensory change. And for this, we're planning on developing a large-scale mobile game of navigation, which will enable us to dissociate sensory change, let's say sensory change from prediction error. So if you have a tunnel that always leads to the haunted house, you'll have a big change, but no surprise, no prediction error. But if your car suddenly won't start, then you have no sensory change, but you have strong prediction error. So to start and tease apart which of these different factors leads to segmentation. And the second two parts use stimuli based on the SIMS, which is a leading approach in our lab, because it gives us the ability to create stimuli that are both naturalistic, they resemble real life, but they're also very highly controlled. So take a look at these two clips at the same time as much as you can. You see, we can create different episodes in which we can change the location, change the object itself, change the character, but we can still have the exact same episode with the exact same timing. And we're using this both to look at encoding. In this part, we're asking whether dynamic events are encoded moment by moment, whether we save each moment to memory as we go along, or whether we indeed create these orbs like in Inside Out. And for this, we combine fMRI, intracranial recordings, which are recordings from electrodes in the brains of epilepsy patients, and modeling to ask whether brain activity of encoding of each element follows that element, or whether it occurs only at the end of the entire event. And finally, we're looking at representation. How do we represent events and their components? And to do this, we're going to be using primarily high resolution fMRI. And here we can use the SIMS to create different episodes that share a character or share location. And now, for example, if a region is sensitive to location, we'll see that the representations of these episodes that share the same location are more similar compared to an episode that, let's say, shares the same actor, but in a different location. So that's very briefly just to give you a taste of the questions that we're asking. The lab is kind of nearly full for the summer, but there are still possibilities to join, especially for people who are interested in staying later throughout the year. And experience with programming is also a plus because it can help you get into things more quickly. But even if you're interested and it doesn't work out this summer, feel free to reach out for next year. And that's it. Thank you very much.
Thank you, Aya. Thank you so much. I love Inside Out, so thank you for nice memories. Okay, Didi, are you here?
Can you hear me?
Hey, hi, good morning.
Hi, I'm here. I'll just switch on my presentation, just a second.
Perfect. We hear you well and we see you well.
Okay. Let's see if this works. Sorry for this. Share screen. Can you see my screen?
Yes.
Okay, so hello everyone. My name is Didi, David Omer, and my lab is focused mainly on using computational tools to study neural mechanisms of cognitive process. Specifically, we study spatial navigation and social behavior, and in this amazing brain structure called hippocampus that Aya just told you a bit about. What's special about us is that we are doing it by using wireless neural methods, neural coding methods, as you can see here on the left. So we use these wireless neural methods to record both neural activity, as you can see up here, and also behavior, in this case in the form of eye movement. And we do this in a very unique animal model, which is common marmosets. So common marmosets are basically a small primate the size of an average rat. So very briefly, we are mostly interested in the main two directions, and the one is what was the neural mechanism for navigation in space and time in the hippocampus. And we know already for quite a while that there are many types of different cells which encode spatial parameters, such as place cells, if you may have heard of, which are considered to be some sort of a GPS system in the brain, as well as grid cells and head direction cells, considered to be some sort of a compass device, which enable us to understand the physical world as well as navigating in this physical world. The thing is that most of the studies were done in rodents, and it's not even clear if all of this amazing neural mechanism exists also in the primed brain. So this is one main question in my lab. And the second question, which is related to this, is the combination between how we navigate physical space and how do we combine this information with social information. And this is very easily embodied in a very simple behavior, which is called territorial behavior. Territorial behavior is basically the ability to combine representation of physical space as well as social information. And we have a hypothesis how this is done in the hippocampus, and it most probably involved a neurohormone called oxytocin, and this is what we are mostly interested in this direction. So very briefly, I can offer two main computational projects in my lab. So the first project involves what is called in our field MRI CT fusion. So the idea is to combine imaging of brains of marmosets, which were done either using MRI or CT. So each modality has its own advantages, and it emphasizes different part of the brain structures. And the idea is to combine this for each individual animal and fuse it with a 3D anatomical atlas of the marmoset brain. So this is one computational project that I have in my lab. The other project, which is a bit more neurophysiological, has to do with a phenomenon called ripples. So ripples are a known phenomenon to exist, to occur in the hippocampus. So it's a sort of a high oscillation event which appears in the hippocampus while animals or humans are either at rest or during sleeping. They are thought to be involved in memory formation and planning. And so one of the things that we're trying to test now is that whether those ripple events basically propagate as traveling waves across the hippocampus. So it's also a kind of a computational project. The data is already there, and we're just looking for people to which are interested and has the skills to deal with such questions. So we are looking for curious and excited students which have also strong programming skills and math skills, because all of the things we are doing basically rely on computational tools. So I presented two main computational projects, but there are many other obvious possibilities. If any of these sounds interesting to you, and even if the summer internship doesn't work out for you in our lab, and you're still interested, please feel free to reach out and talk to us and me. And that's it. If you have any questions, you are more than welcome to email me by email.
Perfect. Thank you so much, Didi. I can see that Ami is here. Good morning, Ami.
Hi.
Hey.
I'd like to, while I'm sharing my screen, then I'll just tell everybody that I congratulate you on taking this first step towards your career in science. And I really recommend that you do visit the different labs and get a flavor of the different atmospheres, different approaches that people take in their labs. It's a wonderful time at the initiation of your integration into the scientific world to investigate and get a broad perspective. So I'll tell you a little bit about what we do. The lab is called the Citri Lab for Experience-Dependent Plasticity, and we're actually interested in a few different topics, which can be summarized under the umbrella of pay attention and learn from experience, which is always a good idea. Our perspective is that the main function of the brain is in defining priorities, meaning that you can do pretty well without a brain, and I'm sure you can think of many examples of people that don't really use it too much, but when you use it, it's there to help you prioritize your actions in the world and to support the prioritization of attention of the information processing here and now. So in terms of attention, you know, your brain is pretty optimized to help you stay attentive to this talk at this point in time and not care about surrounding information that can be distracting. And this is a main aspect of what we're studying in the lab. We believe we've identified a neural circuit that supports this capacity of attention. As you'll see soon, we work in mice and investigate this in mice. The second aspect of the lab is around the selection of actions, which is also a matter of prioritization. How does the brain support the decision of what specific action we will perform based on the information, on the input that we're receiving from the world, which action will we select to output? And this is also the second topic of major interest in the lab. And all this sits on the category of experience-dependent plasticity, which is essentially the mechanisms that the brain utilizes in order to incorporate information from the world and change its functionality. And this can be learning and memory in more explicit domains, and can be the formation of habits, of compulsions, and of addictive behavior, which is something that we spend a lot of time investigating. And we look at this from different levels of analysis. So we go all the way from animal behavior through investigating circuits and systems to synapses, individual neurons, and down to the genetic and protein level, and then all the way back up, investigating the functionality of genes, how they modify synapses, neuronal function, how this integrates into systems, and how this then modifies animal behavior. And I'll give you a couple of examples of what we're currently looking at. So one is we've become obsessed with a neural circuit that supports something that looks like obsessive behavior, these stereotypic behaviors that look like tics or like compulsive actions that obviously have a relevance to human pathologies, and we can mimic them in the mouse. And our entry point to this is looking at drug-induced stereotypic behaviors. So following repeated exposures to cocaine, you have development of neural plasticity that leads to the compulsive orofacial and upper limb actions. And you'll see this in this movie here. So this is, oopsie, this is a mouse. Ignore the mouse on the right. We'll look at it in a minute. This is a mouse that's, I don't know if you can see my cursor. Nili, can you see the cursor? Okay, so this is a mouse that's received a few days of exposure to cocaine. And this behavior of licking the surface obsessively is not natural mouse behavior. It's a overtaking of the mouse behavior that's induced by this exposure to cocaine. And we study the neural circuit supporting it. And how can we actually know that we've identified the relevant neural circuit? Well, the way we can know that, one of the ways is by, and now we'll move to the mouse on the right when this guy's finished his licking. So one of the ways is by mimicking this through artificial activation of the circuit. And this is, you see the laser turning on there. Sorry. This is a different video than the one I was thinking I was talking about. So one of the ways is to inhibit the neural circuit or activate neurons in the neural circuit that are inhibitory towards the action. What you'll see here is a mouse that's received cocaine and is performing these stereotypic actions. So it's licking the surface. Then the laser will turn on and we're inhibiting neurons that we're activating neurons that are inhibitory in the circuit, and they will shut down the behavior. So you see that the mouse is licking this surface. When the laser comes on, then the mouse stops this action and moves about to do other actions. And when the laser will turn off again, which will be in a few seconds, then you'll see that the mouse goes back to performing this stereotypic behavior, which is its major drive. And the laser is off, and you see it's back to licking the surface, which is what it was obsessed with doing before. And another metric to make sure that we understand the circuit is the opposite one, where we stimulate the appropriate neurons and drive a behavior that's very similar to that we see with cocaine, which is this. So you see when the laser comes on, then the mouse starts licking the surface, and then I'll move ahead, it goes off, and the mouse stops. How do we study this? So we use different computational approaches to annotate the mouse behavior. One example here is this use of deep networks to annotate behavior, which was developed by Itai, a computational student in the lab. And there's opening for people who want to continue studying this type of these experiments and help develop this network further. And so here, in terms of understanding experience-dependent plasticity, we're looking at mechanisms underlying the coding of experience, the circuitry underlying the formation of habits, compulsions, and their relevance to addiction. So the mechanisms underlying the development and maintenance of cocaine and opiate. And another topic that we're studying is this brain region called the claustrum. It sits under the cortex, kind of like a sheet, very thin sheet of neurons. Its functionality has been unclear, but in recent years, we and a few other labs have found a number of very interesting functions for the claustrum. So it initially was hypothesized to be the seat of consciousness, where the conscious experience is integrated, the sense of touch, smell, olfaction, visual information, and supposed to give you like the experience of how you experience a rose when you're touching it, when you smell it, when you feel it, when you see it. All of this was supposedly integrated within the claustrum. We came up with more modest definitions for the function of the claustrum in identifying salience. So what is important in the world, as I said in the beginning, how we prioritize information in the world, how we perform selective attention and maintain resilience to distraction, and allow ourselves to modify our behavior according to the environment. And it's also been associated with sleep, which is something we also observe. And the mechanism is thought to be through modulation of cortical synchrony and oscillations, which is a major topic of investigation in the lab currently. And in this context, I'll just show you the summary slide from a project which will be published next week, where Anna and Jonathan in the lab found that the neurons within the claustrum define how a mouse will be susceptible to development of addiction to opiates, to fentanyl, which is a major cause of death in the US currently, and how the claustrum limits the level to which mice will naturally develop a taste for fentanyl, and it allows them to maintain self-control. So who are we looking for to add to the lab? Basically, we're looking for excellent students, and by excellent, what I mean is curious and motivated. Another important aspect is that people coming to our lab, because of the complexity of experiments, we'd want to invest in people that are interested in the possibility of long-term development into a master or PhD in neuroscience. And there is a slight preference for people who are not afraid. You don't have to come with the analytical abilities, but you need to be not afraid to allow yourself to learn analytical approaches and have some curiosity about implementing them. That's it.
Okay, thank you so much. I completely agree. This is like a first step career in the academia, to join one of the labs, even if it's for a short time. So thank you very much, Ami. So actually, we were waiting for another researcher. Mickey London is not here with us yet. So I think it's a time for questions. First, I'll have to say that you should actually approach one or more of our researchers, send your CV, your grades transcript, and like a short motivation of why you are interested in brain sciences, maybe specific in their lab. The decision is the researcher. I'm not involved, so I'm receiving the decision from the researcher. Please take into consideration that the maximum period is 2 months. It can be shorter, and it can be between now and the beginning of next academic year. The scholarships are 16,000 per month, as I said, maximum for 2 months. So if you have any questions, please do. And I'll text also Mickey. I think he can join us. So if you have questions, please go ahead. Don't be shy. You can open your camera or not and ask any question. I can see that Ami is still with us. If you have questions to ask some questions for Ami, I think you're welcome, right, Ami? You're still with us?
Sure.
Sure.
I have a general question about the internship. What, how many time we need to put in the work in the lab? Because it's like how many hours a week, how many days a week? It's a technical question, but I think
yes,
I would like to know.
it's something that will be decided with the lab.
Let me maybe address it a little bit. So first of all, the Milgat Kites, the summer scholarship, is an excuse to get you all here. It's not crucial. If the lab is interested in you and they've already have somebody on the summer scholarship, they'll fund you. This is not a make or break issue. It's just to get people to start thinking about it, to give you additional motivation, and you should be getting some form of support for your work from the lab anyway. In terms of scope, what most labs are looking for is at least a full day, if not 1.5 or 2 days a week over the year. And in the summer, it really is a pretty full-time dedication to science. Otherwise, there's no real point in the exercise. You're not going to, if you just come to the lab and you kind of come a couple of hours a day and you sit next to people that are doing experiments and chat with them a little, it's nice, but it won't take you very far. You won't understand if you want to do this for a master's or a PhD, you definitely won't understand if this is something that you have a passion for. And the lab has no real way of evaluating how you can integrate with them. So the idea of the summer scholarship and of starting over the summer is to allow you to jump in head first, not think about other financial issues over the summer. That's what this support is for. And to really give yourself fully to this experience, and then hopefully also continue it into the year with the lab giving you other forms of funding. So there's normally what students get is a student stipend or a hourly salary that's according to Shat Student is the definition. I'm not sure what it is exactly in a shekel quantity, but it's not far from. I'm not sure. It's not something that you'll get rich from, but it's meant to support you a little bit during your studies and to allow you to devote the time to the lab. You don't work for free. And if somebody asks you to work for free, then you should probably continue your search to other labs. It's not a fair request. Sarah?
Hey, can I ask a question?
Please. Hi, Ella.
Hi, what's up? Okay, what is the main purpose of the research? I just got confused for a second.
My research you're asking about? Or your research?
Yeah, your research.
Okay.
I wish I knew. We're asking a few different questions, and each one has a different objective. So
okay.
If we're looking at the orofacial stereotypies, we're trying to understand the mechanisms of experience-dependent plasticity. So how the brain is modified by experience to perform different actions. If we're looking at the claustrum, then we sometimes are interested in how it defines the selection of actions, and sometimes we're asking how it modulates attention in a specific way. And hopefully the two fit together.
Okay, let's stick though to administrative questions. And
Thank
if you have
you.
any academic questions, I suggest you will email one of the specific researcher you're interested in.
Okay,
So,
thank you.
are there any questions?
Yes, can I ask a question?
Hi.
Hi. Does the program offer a residence?
No.
No?
No,
Okay.
no, I'm sorry. Actually, it's a big issue during the summer. I'm not even trying.
Okay.
But sometimes it is possible, right? Sometimes there are vacancies in the.
They should check independently. I know that it's almost impossible, especially now. It should be really in advance. Mickey is here, so let's switch to Mickey, and if there will be any additional questions, Ami, toda. Thank you. If there will be any questions, I'm here. So Mickey, please, thank you for joining us.
Thank you.
You can share the screen if you want.
Okay. 1 second. What is it? Yeah, here it is. Hi. So, shall English or Hebrew, Nili?
We have some English speakers, so we will continue in English.
All right.
Thank
So
you.
my name is Mickey. It's nice to meet you all. I'll do it very quickly. I have only like 9 slides. Our lab is interested in mainly 2 issues. One of them is dendritic computation, but I'm not presenting it today. And what I want to present today is research that we're doing in vocalization. You know that many animals and humans are communicating through vocalizations, and vocalization is one of the most complicated motor behaviors that we can do. So here's an example of a movie of what's going on around our mouth when we speak. So just kind of for your appreciation, how many things are moving and has to be coordinated in order to produce proper speech. And we are interested in that, in the question of how the brain produces such a sophisticated, complicated sequence of movements. And we're doing that with mice. Apparently, mice are vocalizing. You know that they squeak, but more interestingly, they vocalize when they're interacting with each other, specifically or mostly when male and female mice are interacting. So here's a short movie where you see a male mouse on the left corner moving in a box, and you see a special microphone that can actually record sounds that are ultrasonic that we typically cannot hear. And on the right-hand side, you see the screen of a computer where we analyze these sounds, and we can actually see different type of sound that they are producing. And we and others did analysis of these sounds, and there's complicated structure of these sounds. So it's pretty similar to bird songs in many ways, and to some extent to language. So recently we started to be interested in the sound that mice produces, not only lab mice, but also wild mouse, like the mice that are outside the lab. The reason is that in these behaviors that you just saw, the male mice basically chase the female mice and they are in control. But we noticed that when we bring wild mice and they're interacting, the females are in control, and they basically control the situation and tell the mouse, the male, when it can approach and when not. And we want to compare these behaviors. So one thing that we've done is to record these wild mice. So here you see a recording from wild mice, and you see that in some ways it's very similar to the one that we get in the lab, but the behavior is different. So in order to quantify that, what we build is a system where we have a camera that was taken from an Xbox system, which can actually record 3D movements. And in this system where you can actually put mice in this box, you can put up to 4 mice, and this is what you get. You see, like on the left-hand side, you see how the camera tracks each one of the mice. And on the right-hand side, it actually analyzes the interaction between them. Whenever one approaches the other, if they sniff each other, if they touch, and this is being done, recorded all the time, and can later be analyzed. So the system can actually find many, many types of behaviors and record them together with the video and with the ultrasonic vocalization recording. So what we want to do is to do several experiments in lab mice to quantify how these vocalizations are related to these different behaviors between the males and the females. And later in future projects to do that all in wild mice in collaboration with the lab in Tel Aviv, and also to do it with autistic mice from the lab of Adi Mizrahi that is working on these autistic mice. So to summarize, what you will be doing in this summer project and maybe later is basically bringing mice from the animal facility, put them in the box, and run experiments that we will kind of design in advance, and also analyze the results of this experiment by accessing the system, the database that is being generated during the recordings, and also analyze the vocalization data together with this analysis. So this requires some computing skills. It's not very sophisticated because the system already has a lot of it programmed in, but later on you will need to add codes of your own. And then you also have the freedom to design whatever crazy experiments you want to try that the system allows. And we have many other types of manipulation that we can do and combine for this experiment. So you have like also freedom to come up with a very interesting, crazy experiment, and we'll consider it and you can run it. What we expect from you is someone that is not afraid of mice and can work with them, especially with wild mice that are much more agile. So they run around very fast, they jump. You need to be very kind of on the tip of your toes to handle them. And need some technical skills to work with the hardware and the software, mainly the software because the hardware already done. And if this will come up to work with these wild mice, then some of the work will be done in Tel Aviv, in the lab in Tel Aviv. So you should be willing to go over there. Tel Aviv is always nice to go to, but maybe some of you won't like that. And you need to work independently because right now there are not many people to supervise you in the lab. So on one hand, you'll have a freedom, but on the other hand, you'll have to be independent. That's it. If anyone wants to ask any questions, I'm happy to answer.
Okay, thank you, Miki. I'm very happy you could join us eventually. So thanks again, and Shavua Tov.
Okay.
Any additional questions? Asaf, you're okay? You reached your destination?
I'm okay. Yes. No, we were waiting, but actually, like, I have a few questions. Miki, I'm happy that the system, the hardware, from last year, I remember your talk. Then it was like not completed, but it's good to hear that now the system runs. It's not a question for Miki. Actually, it's a question like generally for this summer project. is there like if you join a lab and then they choose to hire you for a longer period of time, so let's say they hire you for 1 year or let's say for more, so by you doing those projects, is like, can you publish papers from what you do? Like, will you able as a not a graduate student, like will we work on scientific work that will be published? Like, would our names be on papers? Like, this is could be a result from this kind of work.
I think, Mickey, it's a more question that you could answer academically. I'm not sure.
It really depends on which project you join and at which stage it is. And it also depends on the lab. So some labs kind of give credits to everyone that brought water to the PI when he was thirsty. And some labs, you really need to show scientific contribution. But basically, sometimes it happens. So it happened for me that students were on papers, but usually it wasn't like after one summer. So people were here for like the summer and then they stayed for a year. And then like 2 years after they left the lab, they found out that the names appears on the paper that was published. So it can happen, but it's
I guess you need a significant time to join the lab.
You need to contribute, and also it depends on which stage the project is, because if it's only starting, then it takes typically 3 years or more for publication. If it's towards the end, then it's pretty rare that you can contribute at that point, but sometimes it's possible, right? So there's like some experiment that is needed to be completed just for the publication, and then if you contribute toward that. So it's something that you can kind of discuss specifically with the lab that you choose. Yes.
All right, thank you, Mickey. Thank you.
It happens. It also depends on the student, There are students here, right? Lily, I'm not mistaken. I think there are people that were accepted to ELSC already after they had the paper from the undergraduate study.
Yeah.
Sometimes it happens, yes.
Okay, great, thank you. And generally speaking, can we feel comfortable to reaching to more labs that ELSC has, more than the people that we met today? All right.
Yes, of course.
Incredible.
You can contact any one of them, and more than one, as I said.
All right, thank you very much.
Thank you so much.
Good luck.
Bye.
Bye-bye.
Any more questions that I can help you? You of course more than welcome to email me later on if you missed something or any other questions. This is really someone asked something. Hi, Alon.
It's just a technical question
Please.
About signing up to the scholarship. Like, if I'm already in contact with the specific lab, how do I, when and how do I actually sign up for the scholarship?
It should be within like the next few days. I mean, they need some time to review your file and make a decision. The process is that you are contacting one of the researchers, and then they are taking the decision which one of the students who applied to join the lab. It's the researcher decision. Once he or she will make a decision, they will email me and you, of course, and you will know that you are accepted to join the lab for the summer internship.
Okay, thank you.
Is there a deadline for applying the internship?
I didn't hear you well, sorry.
Is there a deadline for applying the internship?
It's until this academic year. So as I said, I suggest it would be within the next few days that you will have the chance to find a lab. If you wait too much, they will have someone already and it will be too late. So there are some labs who already made the decision. There is this program of summer internship, but you can contact a lab whenever. I think this is really important. Don't see it as something that it's not reachable. The PIs really do need your involvement and your curiosity, and you're part of the game. So don't be shy.
Thank you.
Okay. Okay, so have a great week. It was really nice seeing some of you and hearing some of you. Take care, and I hope to see you here. Bye-bye.
Hey, can I ask a question?
Yes.